matrix here, generating an electrical chemical gradient, which is the product of these two forces. It's in part due to the pH gradient that favors osmosis from high to low concentration, and is in part due to the voltage gradient across here. Negative inside, which attracts these guys. Together, they produce this proton motor force, the voltage gradient and the pH gradient. Okay? Which of these components would not be present if these were not charged? If these are just neutral substances, which part of this proton motor force would be absent? It'd be that one, right? Okay, because the voltage gradient here, positive protons are attracted by the negative inside of the matrix. They together create a force that wants these things to fly through that turbine. That flux creates enough energy to spin that rotor and make ATP. In 1961, there was a guy in England named Peter Mitchell who proposed that electron transport chains were generate ATP through a proton gradient. He called it his chemi-osmotic hypothesis. This is the experiment detailed in your textbook that supported his hypothesis. This was done by a fellow at Cornell and at UCSF. Essentially, they made what's called a liposome. They built a artificial cell-like structure made out of a phosphorus bilayer, and part of it came from a photosynthetic bacterium, okay, a purple surface bacterium that has a light-powered proton pump called bacteriorhodopsin. Okay, this is a modified version of the pigment that's in your retina that allows you to capture sunlight and see. Okay, so this is a modified version of that protein, and it's light activated, so it's like the rhodopsin in our rods and cones absorbs light to create a nervous signal to the brain that builds perception of image. In this case, the light absorbed by the protein here allows it to pump protons across the membrane. So they could build this structure, shine light, and then measure the protons inside outside, and they noticed that the thing acted like it was supposed to. The light-powered proton pump. Then the question that they're asking, does a proton gradient, can it drive ATP synthesis? Because that's what Mitchell was claiming. He said the electron transport chain builds a proton gradient that then drives ATP synthesis. So now they've got an artificial system that builds a proton gradient, high proton inside, outside. So then they rebuilt their liposome, their lipid sort of structure. They kept the bacteriorhodopsin present, the light-powered proton pump, and they embedded into it a ATP synthase. With the rotor outside, is on this side of this structure where ADP and phosphate would make ATP if this gradient drives ATP synthesis. They shot in, and I had it in dark and light. So again, outside is ADP and phosphate. In the dark, the pump doesn't work. There's no proton gradient, no ATP produced. When they shine the light on it, the pump pumps protons to the inside, they flow through the rotor, ATP is made. So this supported Mitchell's hypothesis that the proton gradient established by the electron transport chain powers ATP synthesis. Protons flowing through the rotor. Okay, very elegant experiment. Does that make sense? Here's the molecular view of ATP synthase. Okay, this is one of the most ancient enzymes in nature. Everything has it. Every living cell has it. Okay. Now, it might have evolved early in life in bacteria, because all bacteria have the same thing, and this is the same machine in chloroplasts as well. It probably first evolved to remove protons from a cell because it was getting too acidic inside from the cell metabolism. Okay, and so in this case, if you want to pump protons out of the cell, you burn ATP, convert ATP to ADP, and it serves as a proton pump out. Okay, But then if you've got a high concentration of protons outside and in, they can flow down their concentration gradient and cause this to spin and drive the reaction in reverse. I said enzymes go in both directions. In this case, depending on the gradient across the membrane. If I got a lot of protons inside, they'll be pumped outward, driven by ATP energy. If they have a high concentration outside, they'll flow down the concentration gradient, cause it to spin and make ATP out of ATP and phosphate. So the pump can run in reverse, even in your cells. Okay? So early in life, it was probably a proton pump to move protons out of a cell to keep it from getting too acidic. Because remember I told you that in bacterial co-transporters, they pump protons out and let the protons diffuse back in, carrying ingredients with them in symport. We use sodium as the ion in symport. Bacteria use protons. This is the pump that puts the protons out, and it would flow back to a symporter to carry a nutrient into the cell. Here's what I've alluded to already. Okay, here's the symporter that imports pyruvate, okay, and inorganic phosphate to make ATP in the matrix. So here's our symporter. Protons pump into the intermembrane space by the electron transport chain, right? Remember how many protons is pumped into this space as NADH delivers electrons? 10. FADH delivers how many protons in this intermembrane space? Electrons flow through the chain? 6. So the protons are being pumped here by the electron transport chain. They want to diffuse back inward. Well, in terms of our story, we talk about them going through ATP synthase. But they can also go through these co-transporters to deliver things like pyruvate into the matrix. Here's the symporter for pyruvate and protons. Here it is, another symporter to deliver inorganic phosphate. And here's the antiporter that exchanges ADP inside for ATP outward. Okay? And the energy that drives this is just the voltage gradient. ATP is more negative than ADP because it's got an extra phosphate. It's recognized by the positive charge outside, it just moves out that way, the opposite charge attraction. Here, the import of pyruvate and inorganic phosphate is moving from low to high concentration. This is secondary active transport because the protons are flowing down their concentration gradient. Does that make sense? I said a lot of things real fast there, but that's something we've already covered in chapter 5. Remember? In co-transport, it's a type of secondary. Again, this is something I guarantee to ask you on Monday's exam. Can you distinguish between primary active transport and secondary active transport? Primary active transport, an example is the sodium potassium ATPase. It uses ATP energy, that enzyme uses ATP energy directly to pump sodium out and potassium in. And establishes a sodium gradient, high sodium outside versus low sodium inside an animal cell. Secondary active transport uses that sodium gradient, allows sodium to diffuse inward, dragging glucose with it from low into high. That's secondary active transport, right? And here, this is secondary active transport. We've got a proton gradient established by the electron transport chain. In this case, protons want to diffuse back inward. They have 3 paths. They have ATP synthase, not shown, which makes ATP. They've got these 2 different symporters here, where the protons flow down their concentration gradient and drag these uphill from low into high. Okay? I think we're almost out of time here. Let's see. Yeah, we're pretty much, I don't have time to go into this. What we need to talk about after the exam, we'll talk about oxidation of fatty acids, and we'll talk about how your body can adjust your carbohydrate fat storage depending on your diet. You know, people think if I eat a fat-free diet, I won't put fat in my body. No, that's not true. We'll talk about how if you eat way excess carbohydrates, your body can only store so much carbohydrate, and then it's going to convert the rest to fat. Okay. Yeah?
It's chapter 7 on the test.
Jake?
Chapter 7.
According to the syllabus, it says chapter 1 through 6. I'm going to stick to that.
Okay.
Okay. Now the last year's exam, I don't know what chapters I covered. So if there's chapter 7 questions on last year's exam that you have the right to look at on Canvas, of course I'm not going to ask those questions.
Okay.
And as I'm going to say one more time, last year's exam was strictly multiple choice, but yours is going to be multiple choice and short answers. It's not going to be strictly multiple choice. Okay? But at least you can see the level of difficulty of the multiple choice questions. So tomorrow you're turning in lab 8.